treatment using multiple control groups to compare your treatment to. So, and in terms of what those control groups could be, they're actually in the epidemiology and other cancer literature in other cancer types. There's more and more data on these external control arms, and the best data source is considered, patients from large, well-controlled randomized clinical trials, because the collection of data is relatively robust for these large registration trials. So if we look at glioblastoma trials over the last, 7 years, there actually have been, a lot of these large trials, at least, half a dozen of these. So looking at these kind of, I have, 5 or 6 glioblastoma trials. And if you just take the control arm, this is just the control arm, all these control arm patients were treated with radiation and temozolomide, essentially the STU protocol. So that gives you a total of, 1,300, more than 1,300 control patients. And if you superimpose the Kaplan-Meier curves of these patients or these trials, they all pretty much overlap. And you could actually even see the median survival is really close. We're talking about a 2-month difference in median survival in all these arms. And it seems a shame that we have to, each time we do a large phase 3 trial, we have to enroll several more patients, several hundreds of more patients into these control arms. So over the next 10 years, we'll have another 1,000 other patients in control arms just to prove that your new therapeutic actually works. So, and one test of perhaps, statistically, could this, because one thought is that, well, if you use an external control arm, then could you mistakenly, call something that's not effective? And so, this was just looking at some data, looking at, for instance, the EGFR vaccine trial, and then plotting it to the multiple external controls. And that trial here was actually this, the Weller trial 2017, and it's actually the blue line right in the middle. And you could see that, even if you use this external control arm, that trial actually turned out to be negative, was the case when they did the phase 3 trials. Same with dose-dense temozolomide. But the data with the Optune device actually did turn out to be positive when controlled or actually compared to the external controls, which was somewhat interesting. If you kind of do the same for the recurrent glioblastoma trials, it's a little bit more variable because, as I mentioned, when and at what time point patients enroll in recurrent trials are a little bit fuzzier because it's based on imaging studies. But for the most part, if you look at median overall survival, the control arm, which is either CCNU, lomustine, or best supportive care, is still within the range of 5, to 8 months.
Dr. Liao, unfortunately, I've been notified by the CNS that while I think we thought we had 90 minutes of CME to offer, they only offered 60, and so they're really chomping at the bit to get us to wrap up.
So I will wrap up. So in conclusion, how to accelerate the translation of clinical development of glioblastoma immunotherapy. I think we do need better immunocompetent animal models that better recapitulate tumor and host heterogeneity. That's still a challenge in the field. There's different aspects of timing, sequencing, and the role of surgical resection in combination immunotherapies, as Dr. Parney alluded to. And then, the need for better biomarkers of response. And I do agree with Dr. Heimberger that I don't think it's all about the T cells. I think we need to look at the other kind of cell populations that are within the tumor. And then, if we are going to think about using external control arms or more adaptive designs for FDA registration trials. I think that's something that our field really, needs to delve more into in order to get these patients or treatments to patients.